Good afternoon. This is Dr. John Bennett for MedGadget.com. We have another in a series of hangouts for MedGadget. Tonight we're presenting work of Professor Chang from UC Davis on chemically activated HEV VLP delivery. And we have a couple of students that work with him that are going to do the presentation. And we also have Gene Ostrowski, the head editor from MedGadget. Hello, Gene. You're muted, Gene. You're muted.
Hello, John, and welcome everybody to our hangout. And I'm looking forward to discussing how viral particles can be used to deliver drugs now.
Right. This is a new field for me too. Okay, Marie, could you introduce yourself as well as your associate, please?
Yes. Hi, I'm Marie Stark. I'm a PhD student here, working in Professor Chang's lab, and with my postdoc colleague Prasida Halla.
Hi.
And we're here to discuss the recently published nanomedicine paper.
Okay. Okay. Could you tell us a little bit about your background, please, before we start into the presentation?
Sure. Do you want to start, or?
Yeah. So I've recently joined the lab. It's been 6 months actually. I did my PhD from Delhi, India, and my PhD work was on hepatitis E virus and how it enters cells and how it infects hepatocytes, which are liver cells, and using that information here to try and understand drug delivery, especially to cancer cells. That's what I'm trying to do here.
Very good. Welcome. Okay, Marie, you?
Hi, I started out here at UC Davis as an undergraduate, working in my last couple years as an undergraduate in Professor Chang's lab with the hepatitis E virus-like particle. So this particular paper I started out as an undergrad, and then I went on to graduate school and actually have a collaborative PhD right now with Finland, in Uvascular, Finland. So I am doing majority of my work here, though, in UC Davis, and we just finished up this last publication after kind of transitioning into my graduate program, and now continuing to work with the VLP and further its diagnostic and therapeutic applications.
Well, on this particular project, how long have you been working on this one?
Man, I'd say since at least 2011. So.
Oh, okay. For a little while. Okay. Why don't you begin your presentation and just let me know when you want to put the slides up and everything.
Sounds good.
You want me to start with the slides now?
Yeah, that's great.
Okay. Just give me a second. I'm a little rusty. Here we go. Okay. Okay.
All right, so we're going to talk about the chemically activated HEV VLP delivery. So we're going to start with actually talking about the cysteine sites that we chose on the virus-like particle. So if we go to the next slide, so the first aspect of this particular publication was choosing sites on the surface of the virus-like particle. So in this case, we were choosing protrusion domain sites, meaning that they're highly exposed to liquid or the outer environment, as opposed to occluded cysteines within the virus capsid.
Yeah, and the interesting thing about hepatitis E virus and the virus-like particle is that making all these mutations or modifications doesn't affect the ability of the VLP to fold into its proper conformation. And also, having these modifications doesn't affect the shell of the VLP, which remains intact and it remains hollow.
Yeah. So, and that was a method of the design. So choosing variable loops that are more conducive to alteration. So we basically chose different ones and tried them all out, and they actually all happen to still form intact capsids, but we ultimately will be talking primarily about a 573C site that was selected because it appeared to have the strongest exposure upon conjugating ligands or binding ligands. So we can go maybe to the next slide. So to kind of back up, so So first of all, the VLP itself in this particular experiment, we bound surface fluorophores to lysines on the VLP surface, and then we used those cysteine sites that were engineered in the VLP to then subsequently conjugate a cancer ligand or breast cancer targeting ligand or breast cancer targeting molecule, LXY30, at UC Davis Medical Center. This was a previously synthesized, it was semi-discovered and synthesized through a series of libraries that it had high specificity and binding affinity to MDA breast cancer cells. So we actually had that ligand in a collaboration with Kit Lamb's lab at the UC Medical Center. We had that functionalized and bound the LXY30 breast cancer targeting ligand to the surface of the VLP to target breast cancer cells. So simultaneously you have a fluorescent molecule and a breast targeting molecule on the surface so that you can actually track where that VLP lands.
And also because it has a hollow shell, this has implications for both diagnostics and therapeutics. You have this VLP that's fluorescent that can be tracked when you do intravital imaging in animals or in cells by fluorescence microscopy, and it has the capability to target specifically cancer cells, and that's the biggest thing with cancers, having that kind of specificity that you won't have non-specific infiltration into other organs or into other types of tissues. So yeah, that's what we were going for.
Yeah. And actually, if you don't mind skipping to 2 slides from here, so actually number 5, and we'll go back to number 4 in a second. So just a quick idea. So we were actually fluorescently imaging the rat tumor. So what you can see here on the top are the VLP with just the fluorescent label, and then on the bottom you have a LXY30 molecule bound to your VLP and your fluorescent molecule. So essentially what you can see is in applications for diagnostics specifically, you can see that the tumor is much more readily recognized more quickly with this fluorescently labeled VLP at early time points as compared with the top images where you don't actually see the tumor based on fluorescence microscopy any differently. So the idea here is that extra conjugated or bound ligand attached cancer targeting ligand can actually hone in on a cancer target in vivo as well. So that's kind of the ultimate goal, is to have this structurally sound VLP that's already evolved as a virus, this structure, but we've removed all the infectious elements from this virus, and now we're able to functionalize this virus to target cells that aren't necessarily its endogenous cell type. Rather, in this case, we would like to target cancer cells only. And so we can go to the next. We can go right above that then, actually. Yeah, that'd be great. Thank you. So this was just a schematic to kind of give the overview of what is important about this system development. So the idea here is these VLPs are actually produced in a back-to-back system with an overexpression system that's previously been FDA approved. So it's using insect cells to overexpress a protein of interest. In this case, we overproduce the VLP monomer, which self-assembles into a big capsid. And so this process is basically allowing us with very high yield with a relatively quick turnover time. And then we've made this modulatable surface so that post-production, because this is such a stable VLP, we can store it as a modulatable system, and then subsequently as new cancer targets are in development, we can test out different binding modularities and basically test out different systems without having to go back to square one and reproduce the system over again. It's kind of it's a platform that's already made, and then the actual reaction for a lot of these binding is actually like a day reaction or something like that. So that kind of gives the ability for easy accessibility and testing.
Yeah, so this analogy is pretty cool that way. You have the different drill bits and
Yeah. So it's like you can think of a drill, but yeah, I guess for your different cancer ligands. And then we can go maybe to the next slide, past the, yeah. So this schematic is kind of describing kind of what perspectives we're working on now with the VLP. So as I previously mentioned, this is a self-assembling VLP. So the idea it's disassembly in the case of the hepatitis E virus-like particle is is not irreversible like it is in many other virus systems. In this case, you can disassemble the virus and reassemble it. So basically disassembling it into its monomer dimer units and then reassembling it into the full capsid. So in this case, we can actually subsequently package cancer-fighting agents as well. So not only are we thinking about using this as a diagnostic system, but potentially as a delivery system, so packaging drugs, and there's many other platforms that people have tried, but a lot of times you lack the ability to have mucosal stability, so you can't take this orally. Usually you cannot take any kind of cancer-fighting reagent, for example, orally, because any platform will be readily digested in the low pHs, and any liposomes, anything like that, they usually just do not last, and then they also often lack specificity, whereas the case of the VLP, you know, we have evolution on our side where it's already evolved this kind of stability in the mucosal system. And but we obviously had some design in there. And so the idea here is that you can not only specifically target, but then deliver some kind of drug, genetic material, magnetic, metal material, etc. Those are all prospects we're in the process of testing and working on in this current platform. So I think, yeah, so if the schematic, just a brief recap, so the idea, you disassemble, package cancer-fighting reagents, then you attach cancer-targeting molecules to that packaged VLP, then you deliver that VLP to the body. It attaches to the cancer cell that you've already put the targeting ligand to, and then you essentially can then subsequently release those drugs or reagents of interest. And then maybe the last one? Maybe I'll let you. Do you want to talk about the last one a little
this
bit?
is basically a schematic for what we know is happening in the gut. So when the VLP is ingested, and this is important for cancer, especially in patients who are at later stages of cancer, it gets more and more difficult as the disease progresses to have intravenous administrations of drugs and diagnostic agents as the overall health declines. So having an orally deliverable capsule would have logistical and other advantages, and this slide basically shows what happens once the VLP goes in and once it reaches the gut. The tissue of the intestine has specialized cells that are designed to take up antigens from the lumen of the gut, and basically one of the things that we want to do is to modify the surface of the VLP to try and target these gut immune cells specifically, which are then going to get picked up by the systemic immune system and be able to target cancer cells in a more specific way.
So essentially training your immune system to fight the cancer,
Yeah.
which is kind of already a idea in the works, and many people are taking different avenues, and in this case we're kind of using again that already evolved capsid as our system that lasts in the gut, where you have some, or actually the strongest immune response elicited.
So yeah.
So I think that kind of wraps up the current paper or publication, as well as the future prospects that we're in the midst of working on.
Thank you for that. John, you're muted.
As you can tell, we're not experts at this tech. But thank you very much. A very clear presentation. We won't make it a long presentation. Go ahead, Gene. Why don't you have any questions you want to ask?
So, would you say that this is a kind of a novel form of immunotherapy that hasn't been looked into before?
Yes, it kind of depends. I mean, so we've kind of already used aspects, but combined them into a novel system in and of itself.
This is targeted therapy for cancer particularly, but then given the modifiability of the VLP, we can actually use it for immunotherapy, even for vaccine applications. It has endless implications according to.
Yeah. So I think, yeah, and so you'll see, and the other thing to consider, so you'll see the word virus-like particle VLP kind of tossed around quite a bit in biology, and for delivery techniques or vaccine techniques, but kind of the interesting difference between a lot of other VLPs is this is a very full capsid that self-assembles into mono-dispersed units that are all identical to one another. What you'll see, a lot of these VLPs are often actually just small protruding units of viruses that kind of more form what we call aggregates, protein aggregates. So they're not as predictable. They can't necessarily encapsulate. So a lot of the VLPs are formed in this way. Other VLPs that do have that capsid structure, a lot of them are not as stable, so they don't have that pH resistance in the mucosal system, or they're very temperature sensitive. So kind of we have used a virus in this case that already has all of those really stable qualities in nature.
So, can you clarify, like the way you produce these particles? Do you actually take the hepatitis E virus particles, right, and break them apart?
No, that's a good question. So actually, we don't do that. So we take only the sequence that produces the capsid protein alone. So none of the non-structural proteins that are associated with replication and none of that. Just the, fortunately, so some viruses actually require some of these non-structural proteins for their assembly and all of that. But in this case, the capsid structure self-assembles, so we can take a single monomer, overproduce it, and it will self-assemble in the right conditions to a full capsid structure. So we actually introduce that gene sequence into what's called a baculovirus production system in insect cells. So this is like baculovirus essentially is something that only infects or primarily infects insect cells. So you know when you eat lettuce, you probably take in some baculovirus here and there. So it's a very benign system essentially, which is why it's FDA approved.
And it's used for a lot of vaccines for production.
Yeah, it's already in use for vaccine production. So we use that to overproduce our protein, but at no point are the infectious elements ever introduced, so there's no point when it could later on modify itself or become virulent because those materials are not there to begin with for it to replicate and evolve.
Okay.
Do
you expect other viruses, other viruses might have some other characteristics, right? And do you expect this to be transferable to other viruses somehow? Or really kind of
can you clarify? So transferable in what sense?
Well, like this ability to, to use the shell of a virus that, might carry other drugs or can have different attachments attached to it. I mean, will it be easier for you guys in the future to do the same with another virus that has, I don't know, some other characteristics that are advantageous for something else?
Yeah, I think there are many viruses that are modifiable, but HEV is pretty special in a way because the shells of a lot of viruses are kind of flat, and this is something that being a structural biology lab we've learned over the years. HEV VLPs are unique because they have these spikes, and the spikes that are there on the surface, they attach to the rest of the shell through a hinge. So they are sort of independently folding off the shell, and that allows this modification to happen without altering the stability or any other feature of the VLP, and that kind of makes HEV unique compared to other viruses that are similar or that are similarly modifiable.
And their ability to pass through the GI tract, correct?
Yeah,
Right.
that's a big deal.
Yeah, so their stability is kind of one of the strongest points. So adenovirus and virus-like particles are very commonly used, but if you do stability tests, they don't necessarily withstand the same kind of temperature and protease and pH environments that we see with the Hepi.
No, I'm not too familiar with laboratory medicine, but is the encapsulation process done with other studies? Is that a common thing done in the lab where you encapsulate a drug, or is this relatively new?
Yeah, so for the VLP encapsulation, this is actually relatively new, and actually being able to reliably have a single kind of dosage encapsulation, so you can kind of have random association with drugs. Or now lipid encapsulation is not as new. There's a lot of lipid encapsulation that we see often, but usually if it's a delivery, particularly with genetic material, people are usually using live attenuated viruses to deliver genetic material. So this concept of kind of encapsulating genetic material, for example, in a virus-like particle is unique as well.
Mm-hmm. And you may have touched on this, but do you foresee this being used for other medications like antibiotics or something?
Yeah, sure. But I think what interested us was that it could be cell-specific or tissue-specific. So that would be a lot easier to. I mean, that's an important application that we looked at. But yeah, antibiotics, sure. Antibiotics, diagnostic materials, anything that can be encapsulated, because we've seen how this VLP works in terms of being able to encapsulate small materials. Antibiotic delivery, yeah, sure, it can be done.
Well, certainly toxicity is not a big problem with most antibiotics, but there are some antibiotics that are pretty toxic. Chloramphenicol and a couple others that are.
So
you really shouldn't load the patient up. You should give the least possible dosage. So I imagine it could apply to some antibiotics that are pretty toxic.
Yeah, no, that's actually a really good point. And in even looking at how people will get gastrointestinal antibiotic problems where they'll lose their gut flora and have other kind of opportunistic, yeah, that's actually a really interesting point that we haven't actively looked at this point, but that's a really interesting point.
Okay, very good.
One question: is there any possibility of toxicity from the VLPs that might prevent them from, being introduced clinically?
So toxicity, not really. Even when hepatitis E virus infects, it has an acute jaundice-like hepatitis-like disease, and then it becomes okay. What we would be worried about would be an antibody response to the virus shell, but we've done these experiments in the past, and we've seen that modifying the protrusion domain, modifying the surface of the VLP, none of the antibodies that are there in our bodies pre-existing from previous Hepi exposure, they stop reacting to the shell of the VLP.
Yeah, so you essentially are binding to those domains that are normally recognized.
So it becomes immune hidden, essentially. The immune system doesn't see these particles anymore.
And the other point is not only that, but if, let's say, you do have some kind of targeting molecule and you need to have boosters, so eventually your immune system does start recognizing the same thing and then has an immune response. The nice thing about this modulatability is, well, cancer cells, even of a very specific type, tend to have many or several different overexpressed integrins on their surface or specific targets. So in the case that an immune response is inhibiting that delivery over time, if you had to give several doses, you can always change that targeting molecule, and then it will no longer be recognized by the immune system immediately. So this ability to modulate can kind of like mask that particle in multiple ways, so you can actually kind of do a multitude of delivery tactics.
Could this type of study have been done about 5-10 years ago? Or is it only because of the improvements in resolution and machinery that it's possible?
Yeah, I think we know a lot more about the virus and virus structure, and we have better ways, more sophisticated ways of studying the structure, which is why we have all this background information that allows us to make these modifications without compromising stability and other features.
Yeah. So yeah, that's another kind of avenue we're looking at, was actually tracking of this delivery. So this higher resolution tracking is more possible with the new instrumentation and reconstruction techniques that this lab specializes in. So we are looking into those high resolution tracking opportunities with a modulated surface, so getting even smaller and smaller molecules to the surface of that particle to track it.
You know, I've never heard of hepatitis E. I've heard of A, B, and C. But Gene, have you ever heard of E? Hepatitis E? You know, maybe I missed something in medical school, but does hepatitis E cause disease? I didn't know that.
Yes, it's more endemic in China, parts of Africa, and India.
Really? What causes GI? What kind of liver problems, GI problems?
Yeah, and it's an acute infection. It's usually self-limiting. Interestingly, there have been epidemics in the past, but then they're mostly it doesn't have a very high mortality. It actually has a pretty low mortality.
Oh, okay.
Less than 1%.
Innocuous disease. Very good.
Yeah.
Okay, very good. Gene, do you have any questions before we close?
Oh, you're muted.
You're muted, Gene. You're muted. This happened. Hello, Gene. You're muted. You're muted. You're muted.
Sorry about that. Yes. No. This was great. Thank you very much for coming and sharing your research with us. This is really fascinating. I have to say, it's kind of very different than other things that we've written about and we've seen. So thank you once again.
Thank you. Yeah, thanks so much for having us.
Okay, in the forefront of medicine. Thank you very much, guys, for your time. And we'll send you the link. Good night.
Good night.
Bye.